Check, check. Awesome. Thanks, folks. Apologies, a couple minutes late with some streaming issues, but I'm really happy to kick this off. Like, how good is this? This is a bit of an upgrade from last year. Thank you. About Nextflow itself, it often feels like it's about the technology, and there's going to be a lot of talks over the next days, speaking about technology. But when we like to think about it, it's also really about the people, and it's really about how the technology can empower those people to collaborate, to come together, to share. I know that over the last couple of days in the hackathon, we've had an amazing amount of people building some incredible things. I hope we can keep that vibe going to this as we hear some talks. Many of the talks we're going to see lots of really nice and cool applications of Nextflow and some of the technology around that. I think when we think about that, it is also very connected to the science, and it's how we can kind of combine that technology and this sharing of ideas, and then hopefully using some of this technology to help them and enable them to do that. I just wanted to point out, probably one of the better parts of my job is spending time talking to users and really seeing what they're doing with the technology. When I think about that, particularly over the last year, the use cases have just expanded a lot. This is just a really high-level view of some of the things that we'll see over the next couple of days, but also the things I've had the experience to see over the last little while. Obviously, the usage of Nextflow and the ecosystem tools is highly focused in the life sciences, so in pharma and biomedicine in particular. We see a lot of work that's going on in drug development, things like CRISPR screens here, a lot of personalized medicine work which is going on, and be able to use really a lot of sequencing to kind of as a kind of readout on that. Agriculture and animal health, we'll see some talks on that as well, how it can be used for crop development, as well as
user group meetings, giving training, all these things which are kind of happening in the community, and it's fantastic. And so we really want to kind of celebrate that and try and encourage that. So if you're interested, this should be live. I think we merged it like half an hour ago, so hopefully the website will run. If you're interested, go to the Nextflow website /ambassadors, and you'll find a bunch of information about this. We've tried to kind of draw out our vision. You read about it, and if you're interested, you can apply. And this is very plastic at this point. We're kind of expecting, we're learning as we go along as well, so we're really looking for feedback. Yeah, there you go. That's what it looks like. I think and you can see our list of like initial ambassadors there. And there's a handbook. It's very pretty with all the new branding. Okay. Next thing, 2 out of 3. Community forum. Now, as many of you will know, I and others spend a lot of time on Slack. And as I showed you earlier, Slack is kind of growing and growing, and it's fantastic. And there's some kind of niggling things where we think Slack is brilliant for a lot of stuff, but it would be great if we could kind of structure this a bit better, or if we could kind of share this or organize it in a bit of a better way. And so a new initiative that we've just launched is a new community forum. And this is a Seqera community forum which spans deliberately all of the different kind of tool chain that we have that we're increasingly growing. So there's a category in there for Nextflow, of course. There's also a category in there for multi-QC, which is great because we've never really had a good place for the multi-QC community. There's a category for Seqera Platform, formerly Tower, and Wave and Fusion. And so you can post any questions in there because many of the questions we see in Slack span many of these boundaries. So there's a real natural home for that. There's also kind of specialized categories such as events. So anyone can post any events there, and it automatically kind of builds this events calendar. And we really hope that this is a really kind of helpful, useful place for the community to gather. We're at 10,000 people now, and we hope that this forum, it's built with discourse, will help us scale to 50, 100,000. That's the hope. It's a really, really great thing, and offers a lot of functionality here. I love Slack, and Slack isn't going to go anywhere. So we're not going to close Slack down or anything like this. We expect the two to coexist. They serve a slightly different purpose, but certainly for people out there who have a question, want it to get answered, we hope that the community forum will provide that really good structured format for that. And then kind of more discussion type things can remain on Slack, and we keep that community feeling absolutely. It's open now, community.seqera.io. You can go and check it out. One of the things that is the most important thing for me, really, is you can check it out without logging in. There's no invite code, which expires. There's no password, there's no paywall, nothing like that. You can just browse the whole forum. Google can browse that whole forum. So if you type, well, I hope as the kind of library of questions and answers grows here, you can type your random Nextflow error message in and you'll start getting answers from a community forum. So it's a really big shift in that sense. And of course, if you log in with Google or GitHub or anything, you can kind of post questions and all the normal things. So there's a blog post we just pushed out about this as well, which goes into more detail about our motivations and why we're excited about this. So you can have a read of that. And please pop on and pop some questions in. There's a random category. You can post about anything. You can put cat GIFs. there's a Giphy plugin. So get involved and we'll see you there. Final announcement. This one's a person. Many of you at the hackathon will have met her over the past few days, but this will be news to others. I'm really excited to welcome Geraldine onto our team at Seqera, the community team. She's a bit of a legend in the bioinformatics community. We've been moving in similar circles for many years, and she's very well known, especially for her work with GATK, setting up the community and the forums around GATK, especially in the early days, and more recently with Riddle and with Terraform. She literally wrote the book on genomics in the cloud. And she's going to really bolster our presence in the US. So she lives in Boston, and will remain there and be hopefully a bit more available to go to events and things in the States. So she's the new Seqera lead developer advocate, which has been my job title for the last year and a half. But I'm not going anywhere. I'm still moving at Seqera. I'm just kind of taking a bit of a horizontal step over to work a bit more directly involved with product management. So I'm going to be working with a bit more with the roadmap and directions of Nextflow, MultiQC, Wave and Fusion, and of course still very much embedded in the community. So I'd like you all to kind of join me in welcoming Geraldine to our community. We've really thrown her in the deep end because her first day was on Monday. By Tuesday, I had her recording podcasts with people. So, you know, really, really getting stuck in straight away. So I'm really excited to see what we can do together. Right, with that, I will wrap up and I'm happy to take any questions. Thank you.
Okay, we've got a lot. We've got a lot to talk about. We can kind of start off, and I think there's a question around, what do you see the difference in terms of the driver between the pipelines which are released and then all of the pipelines which are in development? Is there like some kind of bottleneck? Is there things that we can do as a community to really help drive those pipelines going from starting in development into release?
I mean, at the end of the day, NF Core is, and I hope basically always will be, a volunteer community. People build pipelines because they need them. People build pipelines because they enjoy building pipelines. So pipelines take time, some more than others. And it's kind of a natural thing that there are often many pipelines kind of in development before the first release. Things we can do to sort of help move things along quickly, the bottlenecks that we've always hit is kind of the very starting point of getting through the initial kind of hurdles of agreeing that yes, this is a good pipeline. We've put some processes in place to help that. And the other one is always the first release, where we have quite stringent review controls, which I think are really important. It's kind of like when you publish a paper, you have peer review. It's the same with NF Core pipelines. When you do the first release, we say, okay, other people have to go and run your pipeline, and it has to work. Things like the AWS megatests and the CI testing we're building up, which runs all the pipelines on other infrastructure, which is not people's home infrastructure, definitely helps inspire confidence. So things like that we're hoping can improve the run-through. But at the end of the day, it's really kind of due to, I mean, we're victims of our own success that lots of people want to build new pipelines. But we just have to do our best to encourage and collaborate.
Next question is around how do you think about chaining those pipelines together? I mean, we saw before we had pipelines, submodules. It feels like there needs to be a layer above that. Like, and how does NF Core think about that?
Good question. Cool. I've had this question since 2018. It's an old one, and it's a very valid one still today. Yeah, it's not an easy problem. There's a bunch of stuff we're doing about it, though. Those of you at the hackathon will have heard Ben stand up at the end of the day and talk about some of the provenance plugins that we have, which are now taking all of the internal metadata that Nextflow knows about the processes and the tasks that have run, the inputs and the outputs, and reporting on that into a standardized file format. So that's one approach which is being actively investigated, is producing provenance reporting and then being able to launch other pipelines from that. Also, I've spent some time talking to Ben and Paolo about how we can sort of encapsulate all the logic about inputs and outputs into pipelines so we can chain them better. It's something we're definitely going to be talking about a lot more next year, I imagine. So I hope that this time next year I can stand here and talk triumphantly about how it's dead easy to do now. We're definitely moving in the right direction.
And then on the ambassador program, we've got some comments around how people should automatically be added to that. Do you want to give a little bit of a little bit more commentary on exactly kind of what we're looking for there and how people can join it?
Yeah, so I mean, I'll start with the second bit because that's easy. How you join is you go to the ambassadors page on the Nextflow website, which just launched, and you read everything, you just decide if you do want to do it, and there's a button, and you fill in a form. So that bit's easy. The kinds of things we're looking for is just basically kind of activity in the community. I mean, the first group of people we've invited, we've just written a blog post as well where three of our new ambassadors, we've kind of talked a little bit about the kinds of things they're doing. Abhinav, Priyanka, and Robert Petit, I think. And they're doing things like running local training courses, running local events, writing content, writing documentation, answering lots of people on Slack. I mean, we don't have we've tried not to make it very strict about what we're looking for because it's a new program and we want to hear from everyone else about what fits. But basically, I see it not so much as like a recruitment thing, but more of like a support program. Like people out there who want to do stuff along these lines, we want to do everything we can to help you.
You'll be happy to know that the streaming has been back on for a little while. So. Awesome. Thanks so much, Phil.
Oh baby, I wanted you to be here inside my arms.
Hi everyone. This is a picture of a microscope. It's from 1738. And it's a gift from the taxonomist Linnaeus to somebody who I actually don't know who it was. And I'm here today to talk to you about something that I think is equally exciting as I think that these 18th century scientists found this microscope to be. And it's about how we can make these kinds of pictures without using microscopes. So this is an image of a cell that we have made using next generation sequencing. And I will explain to you how we do that today. And I will explain how we use Nextflow to do it. Firstly, I'd like to acknowledge that I'm here representing a great team at Pixelgen Technologies and also our collaborative partners. And all of these peoples have contributed greatly to the work that I'm about to present. So, what we study is the cell surface proteome of single cells. And why would you want to do that? Well, it plays a very important role in several biological processes, and this is in no way an exhaustive list, this is just a bunch of examples. So we know it's important for communication between cells. We know that it can work as a mode of action, for example, when a CAR T cell attacks a cancer cell. It's important for cell locomotion, when cells move around in the body. And we know that it's a mode of action, for example, for antibody drugs. And we know that for all of these dynamics, it's not only about how much of the proteins you have, but where they are located. So why use molecular pixelation or MPX to do this? Well, once again, back to the microscope. Nowadays, you can take these wonderful pictures that the people in the 18th century couldn't even dream of, doing these fluorescent microscopy images. And here we see some T cells, where a protein called CD3 has formed these sort of poles or clusters on the cell surface. But microscopy has some important limitations in terms of resolution and also in terms of multiplexing for the number of, for example, antibodies that you can assay at the same time. We, on the other hand, we use DNA sequencing to look at similar or the same things. And we can do this for 1,000 cells at the same time, for 80 antibodies at the same time. And we can do things like this. So this is a uropod, which is a cell that is trying to move around. And then a number of proteins have moved only to one side of the cell, and that's what we see quite clearly here in these examples, where you see the uropod markers on the right. They form very clear and distinct poles on these cells, while the background markers are all over the place. So with our method, you get for each and every single cell, and for all of the proteins in our assay, you get not only how much, so the abundance of these proteins, but you get their spatial distribution, and as well as how they co-localize together with each other. And you do this for 1,000 cells at the same time. So think about what kind of opportunities that really opens up. So how does it work? Well, we start by fixing the cells. So we lock the cell surface proteome into place. We then bind antibodies to these that have short DNA oligos conjugated to them. And so far, nothing is really new. This is when the interesting thing starts to happen, when we then do what we call molecular pixelation. Then we take a rolling circle amplification product, and it goes across the cell surface, and it forms neighborhoods. And we can think of these as sort of zip codes in a city. So you know, for example, which people live in the same zip code. Then we do this for another round, and those connects those neighborhoods together. So allowing us to understand how the structure of the city is actually set up. And you can do this in a standard cell biology lab. You don't need any specialized equipment. You do it in a tube. And we then run this on a standard sort of Illumina sequencer, and you get your data out. And sort of zooming in a bit more on this perspective, we can see on the left here what we are doing in vitro. So on the cell surface, we form these regions, the A region and the B region. And then once we have our data, it allows us to construct a graph, and in this case, a bipartite graph, that then allows us to create a reconstruction of the cell, like what we see here to the side, in silico, for 1,000 cells at the same time. And we are super happy to announce that as of yesterday, the pipeline that we use to do this is released under the NF Core initiative. As far as I am aware, at least, we are the first company to release a pipeline directly into the NF Core initiative, and we are super excited to do that. And I think that these type of maps you're all very well familiar with, and I will use this to explain how we analyze the data step by step. So the first few steps of the process, we basically take the sequencing data in FASTQ format along with some metadata about the samples, and we concatenate the data if it's been run across multiple lanes. We then build our amplicon, depending on what sort of sequencing configuration you have used initially. We then do some quality control on these amplicons, and we demultiplex them so that we get a single file for each sample and marker. So essentially you get 80 files per sample with the current panel. We then go ahead to collapse these sequencing reads into their originating molecule. So we have a unique molecular identifier in each of these amplicons, and it allows us to sort of collapse them down. And then comes the exciting part. This is where we build our graph structure that form where each graph or subgraph in this larger graph is a single cell, or could potentially be a single cell. At this stage, we will also remove edges that are formed due to a very, very low percentage of chimeric PCRs that form as part of the assay. But using community detection, we are able to differentiate those and remove those connections and get back to single cells. In the next step, we go on to find what from all of these subgraphs that we have, which ones are actual cells. And it turns out if we look at the size of these graphs, and you do one of these graphs here, and you find the point where it kind of goes steeply down, you get basically to the number of cells that you put in. And at this stage, we then identify these cells, we compute quality control metrics for them, as well as we establish the abundance of the markers in these cells. And in the next step is where the really special and interesting things starts happening. So here we calculate spatial metrics on the graphs for these cells. So we're able to compute a polarity score, which is essentially a measurement that tells you how either gridded, random, or clustered a marker is across the cell surface. And we also compute the co-localization score. So how often do you find two markers together more often than by chance? Finally, we build a report that you can go in and look at to ascertain if your experiment has been successful or not. And building this with Nextflow has been a great experience for us and something we're very happy to do. So what do you get out of this? Well, you get a pixel file out. A pixel file is essentially a zip archive that you can open and use whichever programming language that you use. We provide tools for Python specifically, and we can also show you how to do it with R. And it contains a number of data frames or data objects that you can read into any programming language you like to go on and analyze this data further. And it also plugs very well into sort of the rest of the single cell ecosystem, being transparently usable with tools like ScanPy and Surat. As I said, we are super happy to be part of the NF Core community releasing this pipeline. Not only that, but we are very interested and committed to open sourcing the software that we do. We really think it's going to improve it greatly. So you can also find Pixelated, the core software that powers all of this, at GitHub and at this address. Finally, if you want to know more, head to our webpage. It's pixelgen.com. If you are itching to kick the tires on this immediately, you go to software, pixelgen.com. You find information about the software, about the pipeline, tutorials for how you can do data analysis, as well as public data sets that you can download and play around with today. And finally, we are recruiting for several interesting computational roles. So if this sounds as exciting to you as it is to me, you should find us over at our booth and talk to us. Thank you.
Thanks so much for the talk. We've got a couple of questions around how good do the antibodies need to be in order for this to work?
I have to pass on that question. I actually don't know, but if somebody is very interested in that, they can contact me and I can pass them along to somebody that knows that.
And then if you think about the proteins that you can quantify in a single experiment?
So we have 80 proteins in our panel today. So if you buy our product, you get 80 proteins.
Awesome. And then when you think about like, what were the main reasons that Pixelgen in particular decided to go down the NF core route as opposed to just building something yourself and keeping it there? What was the logic for going down that way?
So I think that we are all of us sort of very much coming from engineering backgrounds and have in various ways followed, I think, the NF core and the Nextflow community across these years. And I think for us it was almost like a no-brainer to go into this, to make sure that we give something back to the community and can also sort of get all of the benefits back from being part of a larger community.
Got another question around, do you know what you use for the polarization score?
Pardon?
For the polarization score.
What that is? That is a Moran's I statistic that we compute.
And then how do you think about the difference between the spatial proteomics and spatial transcriptomics in terms of applications?
So I think that there's most spatial assays today, they focus on tissue. And this is very different from that, of course, in that it's working on the surface of single cells, not on a tissue. So the applications, of course, vary depending on what type of material you're studying. And I should mention that for our product, it's also sort of based for PBMCs, the human immune cells. That's the first product that we have out now.
Cool, awesome. Thanks so much for the talk.
Thank you.
So now we have a break for about half an hour or so, and we'll see you back.
Oh baby, I wanted you to be here inside my arms where the night goes down. Oh baby, I wanted you to be here when the sun comes up and the day goes by. Oh baby, I wanted you to be here inside my arms where the night goes down. Oh baby, I wanted you to be here when the sun comes up and the day goes by.
Hi everyone. If you could please take your seats, we're about to start the next session. So I'm very excited to be able to moderate this next session. We've got two fantastic speakers. I have to say that really, but it's true. First up, we've got Evan Floden from Seqera, who's going to tell us all about the latest developments going on at Seqera. So Evan, over to you.
Great, thanks so much, Phil. We've been doing a bit of soul searching, thinking about what really made Nextflow special and how that connected, how we can apply that in other ways. And what we've really settled on, what we've sort of been thinking about is really kind of three core concepts. The first of it is around open science, around the idea that people can come together, work on a single repository, work on a single tool, have domain experts that can all come together and really kind of catalyze innovation via that. I think it's where we see the growth of NF Core and Nextflow. It's really kind of testament to that. And it's something that we really want to like lean into as a company, and something that we see as something that's really important as part of our values going forward. We also think about the kind of concept of scale. I remember the first time I ran a Nextflow pipeline, I was at CRG, and you just sit there and you type in, you say Nextflow run, and just hit the cluster, and then you get an email from the sysadmin, and like you've just absolutely blown everything. But it has this ability to really, really run things at scale. And we were doing that previously in HPC, and this is kind of something we continue to do. But when we apply that to the cloud as well, it's just incredible what we can kind of see there. The challenge that we all have today as scientists is actually how we analyze so much of that data. How can we do that and make it accessible to so many people as well? So something that we're really thinking about is how we can continue to push that scale across really many different sorts of applications. And finally, I think that the thing that we maybe speak most about from a technology perspective is flexibility. And this concept that we can take modern software engineering tools and really apply them to science. Really take those tools and make them applicable to the kind of work that we do. And I really think that that's a lot of the essence of what we've done. So this process, I guess, we've gone over the last year is trying to understand what that essence is, distill it down, and then kind of use that to apply to a lot of the other work that we do. Going a little bit more practical here, of course, like the idea of connecting into any compute. The fact that independent of where you're running, if you're running your pipeline in AWS, in USS1, or you're running your pipeline in Germany on an HPC, or you're just developing locally, that you can do all of those things and you can really have that same developer experience. Concept of connecting your data, your storage, and this is something which is becoming increasingly common, that the data is spread in many different locations. The cost to move the data is obviously extremely expensive. The fact that you can spin up resources where the data is becomes incredibly valuable, but also you just want to connect into it, provide visibility on it, and connect into where those resources are. For us, containers are really a core concept. It's something we really pioneered back in 2014, saw how they were being applied in terms of long-running web services and how we could apply them to data pipelines. And in doing so, it was a sort of a challenge that everyone had in terms of running these pipelines reproducibly, portably. But in saying so, we can also take that and now apply it to many different technologies. And this is something we'll speak about, and which Paolo will speak about later on. And finally, the concept of source code management, that there is a single repository that anyone here can have permissionless access to download, to view, to contribute to. And it's that kind of collaboration which allows domain experts to kind of come in and do that. And these are kind of modern software tech concepts, but we can apply them to science. And when we apply them to science, we really end up with scientists kind of working in this way, empowering them, and then for them to be able to also apply that technology to broader groups as well. We've settled on this idea of the modern biotech stack. Now, it kind of sounds in some way that it's very precise, it's very prescriptive and things. But when we think about it, we think about the concept of modern and modern software engineering, the idea of applying modern technology, and a more broader concept of biotech, not just the simple application and thinking about biotech in terms of drug development, but really about how we can combine technology and biology together, and then using that for more general advancements of health, of food, of environment as well. And I think one thing that we've leaned into a lot is the concept of a stack, that the tools that we all work with are multitude. They're various. There's many different things. There's no one size fits all. And in doing so, being able to really provide folks and to give people that full stack helps a lot in terms of being able to give them the flexibility, mix and match, and something that was kind of more of a modern software engineering approach. Summarizing that and thinking about what we do with Nextflow. So obviously Nextflow has been around a while, but it's really providing an anchor here. An anchor is providing those scalable scientific pipelines, and also kind of building on the idea of using containers, running things in a distributed way, which is not always easy to do, and making it possible for folks to connect those pieces together. When we look at other things and related way, multi-QC has a kind of open science component where people build plugins, they build essentially extensions for particular bioinformatics tools, and then they can then be used, reused, and applied to those pieces. Slightly lower in the stack, we can think about container provisioning, and Wave in particular is incredibly important for thinking about dependencies. When you look at a pipeline, it's often made even from a single process, 10s or 20 dependencies. Managing that, while it's fine that we can define these things for a pipeline, let's say, we often have many, many containers per pipeline. Managing that across different infrastructure is not always obvious. And when we think about the cloud and the storage of the cloud and efficient sort of computational storage, Fusion plays a big kind of piece in this. So last year, we introduced many of these tools. We introduced this stack and how it can apply. And now we're really going to go a little bit deeper and see how we can use those tools and how they can kind of come together as part of what we're doing with Seqera. So many of you know we previously had Nextflow Tower. So Nextflow Tower has been renamed into Seqera Platform. It's really a single platform that combines many of these things in a way which makes it easy. It's a kind of a way that you can deploy this full stack application. You can go all the way from data generation through to insights, and also having many different ways which you can interact with it. I think from a very simple perspective, you can think of Nextflow as a single user command line tool. It doesn't have a database. It doesn't have an API. It doesn't have a UI that you can interact with. Really by building those things around that, which is what we sort of started with, we realized that we can sort of leverage that approach and apply it to many different sort of other applications, which are very adjacent. Pipelines are a difficult problem, but they're not the only problem. And that's something that we're sort of been thinking about more recently as well. Built, though, on the tools that you use. So building on the infrastructure that you have, whether that's in HPC and cloud, whether that's you use GitHub or GitLab, whether that's that you're connecting in with container registry X or container registry Y. For the way that we build it, it's really a way to integrate all of those tools for you to be able to use them and kind of bring all of that together. Of course, there's a key component of this, which is the open science foundation, the idea that you're building content, you're creating pipelines. You can take something from NF Core. You can develop a module in multi-QC. You can build a container from biocontainers and bring all of that together into the way that you need to do it and kind of continue to leverage, in some sense, the kind of community resources there. Finally, what we're trying to do, some of us are really kind of in the production setting where we're running the same thing often and often. Many of us are also developing pipelines which we want to give to other folks. We want to make those pipelines available. We want to make those analysis available. And the key thing here is kind of giving people an option on how they do that. A lot of that comes from the kind of interfaces that we do. So Nextflow command line tool, we've been building UIs, APIs, and trying to make things a lot more accessible for a whole bunch of folks. Now, I think it was great we saw the talk before, what they've been doing with the Australian Biocommons there. A great example where even within one organization, it's got a deployment, it's got installations in clouds, and actually in all the clouds, in HPC, kind of running everywhere, really means you need that flexibility. That flexibility is something which is just sort of essential to run in this way. And it also means that when you deploy the application, you want to be able to deploy it on enterprise, in your HPC, in cloud, and then connect into those resources as well. So something we're kind of coming from an open source background. It's kind of very important to think about that, but we just see the demand from users and how they want to run the application. It's very important to do that. I want to jump now more into the kind of use cases that we see for pipelines. We've focused a lot on here, on this kind of, actually you can see my cursor, on the kind of concept of like running these really high throughput pipelines, really data analysis pipelines, batch computation. That's really one piece of the puzzle. It's a difficult problem. It's distributed typically. It's something that you need to run very long, but it's really one piece. What if we think about the other parts? How do we write tools and software? How do we run those analyses? How do we interact with the results and then really kind of generate data? And often it's a cycle. It's a cycle that you're going through. Maybe you're more in a situation where you're iterating, maybe you're more in production, but all of these pieces are really valuable. And that's something that we've been thinking about, how we can apply and basically make Nextflow pipelines kind of more accessible in this way, kind of across that whole data analysis lifecycle. I'll just give a quick recap on what's been happening with pipelines. So obviously concept, Nextflow pipelines, scalable, really connect them to your tools and run these things in a collaborative environment. So the concept that you have an application which you can run them in. We look at a quick overview of what's happening there, obviously connecting into cloud, HPC, Kubernetes. We have developed a way where you can have custom user interfaces. So you can create drop-downs, you can drop UIs, you can create any kind of front end that you want for that. We've also been consolidating these diverse computing resources. So from the example before, independent of where you're running, you've really got that single place that you can find all of that. When we look at what we've been going on the last couple of months, it's really about making those pipelines a lot more usable in terms of the details, really treating pipelines like a first-class citizen, a little bit like Nextflow treats Git repositories, linking them to runs, importing and exporting, really treating them in a way which can be seen. The costs and the budget control is also a really big one, something we've been focusing on a lot, how you can track costs, but also associate particular costs to a particular run. And as groups expand, a lot of the hesitancy in using cloud comes from folks who don't feel that they can have control of those kind of cloud budgets. And making that possible is a kind of big goal of what we're working on, particularly when you see the kind of academic use of that. Optimization is massive. We have customers and users who spend $10,000 per execution. So the amount of compute which is going on here is massive. If anything that we can do to improve that, whether that's through making the storage more optimized with Fusion, running things on Fargate, so you get faster startup times, you get quicker results, and also kind of multi-architecture loads, things like AWS Graviton and things that are coming along. The other thing is really about making the whole environment replicable. We think about an Nextflow pipeline, you can just say Nextflow run, drop your repo. Really applying that same concept, but not just to the workflow execution, but really the whole thing. So can you say, I've got this workspace and it's got 10 pipelines, and each one of those pipelines is represented by a JSON file which I can import and export. And Harshal's going to give a great talk on this later on, where we basically say how we can replicate the whole research environment, make everything sort of work in that way. So I'm going to jump into a demo now. I give you a slight warning that this is going to be a slightly edgy demo in the sense that I have many new parts going on, but we'll jump in anyway and go from there. So I'll start off with giving you an overview of what we can do with pipelines. So here you can see we're logging into the Seqera platform. Once I log into here, you can see I can log in. I have a view of different pipelines that are available here. We're going to run through an experiment today. We're going to do an experiment where we're going to search in CBI. We're going to find some samples that we want, based on the accession, and then we're going to upload them as a sample sheet and really download from there. So in the first case here, I'm going to go to NCBI, and I'm going to search here for this particular search query where I'm looking for these RNA samples that are not a cell line, and really find what's available in public resources here. And when I do so, you can see that I get a search, I get a series of samples which I can select. And obviously, I can query these, I can kind of go deeper into these. But by selecting them in the first case, all I'm going to do is download the accession numbers. I just really want those SRA accessions, which we're going to use as the input to a pipeline. So I'm just going to save those as a file here. And then in doing so, I'm going to get a file here. And that file itself, we can kind of interrogate it a little bit more. But in particular, I've just created what we call like a sample sheet inside of here. And inside of that sample sheet, we're just going to have some columns, really like CSV file, very simple, with some description. So just a quick kind of recap on what we can do here. We have a concept of a launch pad where we can launch pipelines. We have a concept of runs where we have history of those workflows that have been run. We have a data sets I showed you before, computer environments. I think the real value here is that I'm in one workspace, but I can run that compute really wherever I have a computer environment, whether that is running in my AWS Ireland environment, or running this in Azure in Virginia, or whether I'm running this in Helsinki, in Google. All of this can be really built into here, and it allows me to have the same pipelines, the same code, and really just change out those computer environments independently. This is particularly important. These examples may look a bit extreme, but imagine you have a development environment, a test environment. Imagine you have multiple AWS accounts which you work on. Imagine you're in different regions. The ability to standardize much of this infrastructure is really important. So we have our data set, which we've uploaded from before. We can see that when we go to run the pipeline here, we can go create a launch. I'm going to choose the fetch NGS pipeline. This is an NF core pipeline. And this pipeline takes accession numbers, so I can literally just feed in what we downloaded here. I'll just give you a quick look of what that looks like. You can see we're pulling from NF core. We're running a specific version. We're running 1.10.1. We've got some labels on that, and I'm going to launch that pipeline here. I mentioned before about this customized UI, which is available. So this is something which has been developed and is part of the Nextflow schema. You can apply a name. You can find the input. So in our case, we're going to look for the input that we have. But I think the really cool thing here is that we can define what we want the output to be as well. So we can say, hey, I want to run after this RNA-seq, or I want to run after this ATAC-seq. So in this case, I'm going to find, I want to run RNA-seq pipeline, choose the RNA-seq here, and then it's going to show for me all of the data sets that we have uploaded, and we can launch the pipeline from there. In this case, it's going to take the individual samples, it's going to get all the sample sheets, it's going to pull the FASTQ files, and we're going to be able to launch and run from there. I'm going to, instead of sitting here for 20 minutes and watching this happen, I'm going to show you one which we completed earlier. It's a little bit like cooking show. So I'm going to jump into here, the fetch NGS, and this is one which we ran earlier. And here you can see that the command which has been launched, we can see that we have parameters, configuration, and any data sets that we launched. So we're linking back to the original data, as well as the execution log. But most importantly, I want to show you the reports. This is the output of that pipeline. And remember, this is just downloading files from wherever, in this case SRA, and giving me a full sample sheet with all of the metadata. So now I have the sample. I have the names of those FASTQ files, the strandedness, I have the accessions, whatever was in that information here. So I know I have all of that bound to the sample sheet. And the concept of sample sheets is pretty simple. It's CSV, TSV files, but if we use them in the correct way, we can actually get relatively far. A little kind of quick touch on some of the things I showed last year and how they've improved. Well, if we think about the optimization of pipelines here, here is an RNA-seq pipeline, which we've run before on human samples. We now have the optimization. So I can define a machine learning model based on an execution. In this case, I'm looking at the RNA-seq pipeline, which Dr. Patel ran a couple of days ago. And I can see I want to determine what the CPU is going to be, what the memory is going to be, and then from that run can determine a configuration profile. Lots of different tasks. Obviously, doing this manually is obviously a significant step, but it obviously helps. It helps a lot there. Okay, one demo down. That was actually pretty good. Okay. Kind of onto roadmap and how we think about that. Well, obviously tracking changes within the pipeline details is really important. We want to bring the same versioning that exists within Nextflow pipelines. We want to reduce the wasted time on validation. Like in this way, when you launch a pipeline, It still needs to run, and then only when it runs do you get this validation step, do you understand what's happening if the data exists, etc. So really removing those steps to make things much more efficient and kind of giving those results to users earlier. We also want to be able to make the pipeline much more interchangeable. So at the moment, how can you take a pipeline and say, I want to give it to person XYZ independent of where they are, using Git as well and kind of making the pipeline more of a Git object, and then really the tighter integration with data. And that's kind of what I'm going to go into next. We have kind of in some way just given the data as being like, it's a, this is something we leave to the reader to make up in some sense, and it's something that's always bugged me a little bit, and I'm very, very glad today to be able to announce that we've really made huge steps and huge strides in solving that problem. So today we're announcing the public preview of a feature that we're calling Data Explorer. Data Explorer has the same concept of Nextflow in that it connects in with your buckets. It connects in with the storage that you already have, but it provides this ability to visualize, to search, and to manage that data. So this is available in a public preview today. So if you go onto Tower Cloud, you'll find that. Giving you a quick overview of what it does, well, it really looks across those clouds. So I mentioned before, if you've got different accounts, different cloud providers, different regions, it provides you that view of that. It has this ability to link the pipelines and the data together. Super important for troubleshooting, if you want to dive into a work directory, for example. We also have the ability to browse and to download and to upload. The object storage and requiring that every scientist has access to your AWS console, no offense to AWS consoles, but it's probably not what we want to be doing in the kind of long term. And so providing this interface that enables the scientists to do that really reduces a lot of time as well as costs. And finally, that area of like linking in public data sets. We strongly believe that many of the public data sets are moving to cloud. I actually remember very early conversations I had with Paolo in 2015. He said, one day it's just going to be there. And like, it's true. The data is just going to be in the cloud in some sense, and it already is in many ways. So providing this concept that we will be linked into that data, and I can kind of show you how that works as well. When you think about it, it's linking in with the S3 blob storage, Google Cloud, so very much similar to what we do already. It adds buckets automatically, so whatever credentials that you have in your workspace, it's going to use those credentials. And really, the access to those credentials, really the access to those buckets, is driven from those credentials. So system admins of cloud have full control over what's going on there. And so we're very excited to launch this today in Seqera Cloud. So we'll jump into demo number 2 and see how we can go with here. So jumping into Data Explorer, that last example, which I just ran previously, if we want to imagine what we did there, we just had our run. And if we go into that particular run that we had, there was a report which was generated. That report, the result, was a kind of a data set. It was a CSV file. But what about the underlying files? What about like, where is this file actually? This is just a metadata or associated data, I should say, on top of that file. How can we go into that? So you can see here, this is in the Seqera Lab showcase bucket. If I was to close this here, I can now jump into the Data Explorer. Inside the Data Explorer, I can see all of my buckets that are associated with my credentials. So you can see I've got some AWS bucket here in EUS1. I've got some test data in EUS2. You can see I've got some Google storage buckets, etc. Really, everything is accessible from a single interface. If we go down into the Seqera Lab showcase, I can find that exact run I had previously, which was the NF Core NGS run. Inside of that, I can find particular results. I can jump in there and find the sample sheet, exactly like I just showed you a minute ago, but I can also go and show you the original files as well. So in this case, I could download this, but I can jump in and find, for example, the actual FastQ files. So it provides a really kind of easy way to navigate, to integrate with your actual results, where they are, and kind of have that much more kind of native experience with that. That's like one direction. What about the other direction where you're like thinking about what files did my pipeline create? If I have a particular run, how can I access and view those files? So if we go to that same run now, we jump in, I can go and find the work directory, so I can basically link into like what happened. Super useful for debugging. If you're familiar with Nextflow pipelines, they create these work directories. Being able to jump in there and see exactly what's happened from the workflow level is important. You can even go deeper and you can say, show me at the task level. So I can jump in to the task table. So this pipeline, just recap, 34 tasks, 34 containerized executions. Each one of those executions has got its own log, it's got its own data explorer. I can jump in and find those files and see them there. So it just helps a lot with sort of many aspects of managing those pipelines as well. How is this kind of driven? Well, I mentioned before it's kind of coming from, a lot of this is really coming from the credentials. If I look at those credentials, I can see I've got Azure credentials, AWS credentials. I can kind of, whatever credentials I put in there is going to automatically load in. But I can also kind of go beyond that and I can start to add public resources. I think this is where things start to get really interesting if we think about the cloud as the kind of default location for many of these things. Jumping in and jumping back into the Data Explorer here, I'm going to now search for example, 1,000 genomes, slightly old, but it's been kind of a classic. So we can search for that. We can find the 1,000 genomes bucket. And in this case, it's really just an S3 bucket. So that means I can copy the name of it. I can take that bucket there, and in this case, I'm just going to jump back into the Explorer. I'm going to add the bucket as a AWS public bucket, and then I'm going to give it a name or whatever, give it a public thing. Press add. Cross fingers. And then if you can see that now, just with that one thing, I have access to all 1,000 genomes data. I can simply like take that in, bring it in, and load it anywhere I want. It kind of goes further than that as well. I can do the same thing with reference data. Here's some data from our friends at the Broad. They can see that here we can access all of reference data. I think the thing I'd like to stress here is, though, that this is moving the workload to the data. So that data still exists there, but I can now run my pipeline, or I can run my analysis where that data is. And that's the really key thing, because no one wants to move that stuff around. That's very expensive. But if I can spin up a computer environment in that location, like I've done here, or like I could do here, then I can simply access that data and kind of run it in that location. There's a couple of other things we have. We've got upload and download, really kind of the ability to kind of take that data as well, link it with other things, but it's really kind of a first step for us, and just linking, really making that connective tissue between pipelines and data, and something we've been looking to do for a while as well. Okay. Where are we going for the roadmap for this? Well, the first thing is kind of obvious, like linking the input and the output with that. So basically making it easy when you select the pipeline you want to run, which data do you want? The validation of that's going to be really important as well. How we can search for more public data sources. I was showing you buckets, but there's no reason we can't do the same thing for SRA, for things like benchling, for base space. We also want to support shared file systems, things like NFS, HPC systems, and really kind of data packaging and provenance, and kind of looking at some of the kind of ecosystem products around that, and what can we leverage that exists, that users are already using, and kind of applying that to this as well. And this kind of takes us on to the next step, which is, this is kind of we thought about like the data management piece, but if we think about how we can make those sort of analyses, that data accessible to many people, for everyone that runs a pipeline, it's really my view that there's 10 people who want to run a notebook. There's 10 people who just want to touch that data, who just want to interact with it, who want to write a small script, who don't want to spin up something really big. And when we think about that, that's something like, whether that's the end of a pipeline, whether that's the really development of the tool that you want to do, this is really what we've been thinking about in terms of what we call a data studio. So just a kind of quick recap. Data Explorer is now available today, so if you log into Tower Cloud, you'll find it available there. You can log in and access it there. It's going to be released in version 23.3 in about a month or so's time. Going beyond, say, that batch piece onto the interactive and how we can think about that, also very excited to announce something that has also been a long time so in the wings as well, which is around a product we're calling Data Studios. And Data Studios is available now as a private preview. It links the ability to create notebooks, interactive applications, where your data is in the same environments that you run Nextflow. The concept here is that we can really take RStudio, Jupyter Labs, VS Code, really anything that you're currently familiar with using, taking that downstream analysis from the pipelines or debugging pipelines or really developing new things and connecting them in where you run that things as well. I think the really nice thing about this is that when you're developing a pipeline often, you kind of need the real data. You need the real environment. It's all fine and it's great that we develop locally and we can try and make things as standardized as possible. But in reality, sometimes it's just being there in that environment with that data makes all the difference. And particularly when we think about mounting sort of cloud data and data around, it's something that can help a lot. When we think about what Data Studios and how it works, we leaned in a lot to the container here. We have 10 years experience of running Nextflow containers and running Nextflow as a container as well, like it runs in platform. How we can think about the dependencies. It's really, I don't know if you spend much time in RStudio, but like defining the dependencies of bioconductor, for example. Extremely painful thing, particularly if you want to make that replicable. How can we learn the lessons of Nextflow, reduce the challenges that exist in terms of defining dependencies and things like Jupyter and RStudio, and kind of take that technology and give it to a whole new set of users. We want to make them portable and shareable and collaborative. Really just like the same way that a Nextflow pipeline, you drop a repository. How can we do the same thing with interactive environments and really have that kind of access control through Git credentials? Okay, this is the most risky one. So we're thinking about how that looks like. So we now have Data Studios, which is available, say, as a private preview. You can add a studio exactly like you would almost create a pipeline. So I can choose the studio that I have. The future is, of course, to choose really any image, whatever image you think is relevant. In this case, I'm going to choose an RStudio environment. I choose a computer environment that I wish to run. And I think this is the real key. This is what makes this a little bit magic, is the concept that I can mount whatever I want. So I can take, for example, my 1,000 genomes bucket that I had, or I can mount inside of here reference genomes that I need, or really any data that exists, the output of my pipeline, for example. And when I do that, I mount that data inside of here, and that data studio which gets created, I can use that data in a way. I will show you some kind of what this looks like in a couple of examples. And I think these hopefully are kind of illustrative of what we can do with here. So first in RStudio, you can see here that I have really like an R Markdown file. This R Markdown file is actually taking the output of NF-core RNA-seq. So NF-core RNA-seq creates a file, which is an expression file. And what I can do is I can read that file in. But importantly, notice here on the right-hand side, you can see it okay, but here's the data. This is really mounting the bucket that I had inside of there. And I can mount the bucket inside of the container, so inside of that environment, and then run it in that way. So here I've got an expression matrix, which I can generate. I can run my RStudio code in here, create that expression matrix, and then generate my graph. I'm going to go to another slightly, I guess, slightly more new age example with Jupyter. Likewise, the same thing. I've mounted the data inside of here. Now I'm going to create a pandas data frame from the same data. So it's a TSV file with genes as well as samples and then the expression of those genes in that file. Just going to launch that. I'm creating a data frame. I'm going to load now the Jupyter AI module. What this does is it allows me to feed data into my data, the data I've loaded from Fusion, into here, and I'm going to pass that to OpenAI. And in this case, I'm going to ask it to write a script for me, and that script is going to say, take the output of this tool, which is a salmon tool from gene expression, from NF Core RNA-seq, write a script for me that's going to generate different figures that are commonly used for differential expression analysis. So when I launch this now, this is going to take that input full, pass it in here, run that against that. The data, the original data here, though, is browsable from here, so I can access that. It's really the output of my pipeline. And from that, it takes a couple of seconds to load through. I just give that a moment. And in doing so, it's going to generate for me, hopefully, it's not exactly reproducible. It's going to generate for me a script which I'm going to be able to run and generate some figures from that. Kind of an example of how we can really combine things together and perhaps to use this concept of particularly in AI. Yeah, here we go. I just want to point out here, it's very specific. So it's really determined the different conditions which are available here, kind of gone in and being able to generate that as well. If I was to take an example of that and run that, you can see we get gene plot expressions, we get different parts, we get an expression plot based on that, and we can get different things. So lots of kind of interesting things. I'm going to finish off with like the last part of Studios, and this is really about not interactive analysis per se, but really about development. If we think about how we can develop those pipelines in that environment, we also have available VS Code. Now, I know there's like many extensions for this, so what we can do here is we can pull in our pipelines, develop those pipelines in situ, in the environment where we have access to that information. And really close off, I'm not sure if it's a complete term, but there's interactive development environments. I really think we're creating interactive scientific environments here, things that people can build on, people can use, and kind of go from there. Okay, kind of if we think about sort of roadmap from here, as I mentioned before, dependencies. This is a very difficult problem, something we really want to work on. Replicating those environments with snapshots, supporting different clouds as always, supporting HPC, supporting Kubernetes. It's really, well, as working with the community. Nextflow became a de facto standard, but it wasn't intentional. It was something that came out. How can we do the same with these kind of environments? What do people want to define their dependencies as? There is a couple of standards which exist, things like dev containers out there. Are we going to build on top of that and kind of use that to be able to build interactive scientific environments? Finally, this is available today, so in terms of a private preview. If you go to this website, put in details, we'll sort of contact you to make it when it's available. Would love some feedback on this as well. It's really kind of new stuff we've been building on. Ideally, kind of completing this, it's still very early days of what we want to do. And as we kind of think about completing this whole data analysis lifecycle, something that we see that there's a whole lot of potential to close off and just really make much of this stuff much more available to folks. Just kind of point out on how this is available. We have Seqera Cloud Essentials. This is a completely free product, which you can do. You can sign up for Seqera Cloud. It includes pipelines, Data Explorer, and Studios across all of the clouds. Limitations are around just the number of people that you can have in your organization, as well as workspaces and concurrent runs. We also have a new academic program, which we are launching, which is Seqera Cloud Academic. So this really gives you all of the fully functioned Seqera Cloud Professional for academic institutions. So you can apply to this and you can tell us what your organization is, and we will kind of give you fully loaded on that. Just a reminder, there's cloud, there's enterprise. You can deploy this where you want. Independently, though, the computer environments are kind of running there. So it does give you a whole bunch of sort of options on that. And then just kind of finishing up, what I would love to see in like something that's relatively, it's an evolution, but it's something which has come to me more relatively recently is kind of thinking about the infrastructure that we build, but not just the infrastructure, the resources. When I look at NF Core, the value is really that content. It's really the people building incredible stuff. And how can we replicate that in other places? How can we do the same thing for software, like biocontainers? How can we do the same things for data? How can we do the same things for interactive environments and studios? So I'm really looking forward to being here in a year's time and maybe figure out some of those problems. But in the meantime, I really want to thank so much everyone for the time. Hope you can see we're really excited. Obviously focusing a lot on these things, and come talk to me during the rest of the couple of days. Would love to chat. Thanks so much.
Thanks, Evan. I should like to point out, I saw a quick preview of this on Sunday, and I was thinking to myself, this is really impressive, but I'm a bit scared. So congratulations for pulling that off with those live demos. That's good. As you might expect, lots of questions coming in about this. Hats off especially to Anthony and staff who came in early with some questions asking about whether it might be possible to browse files in Tower and download them, maybe even launch Jupyter Notebooks one day. So yeah, just to beat you to it almost. One of the things, one of the questions here from Philip, is it possible to integrate an external LIMS API to create a web app where one can choose samples to process in Nextflow Tower?
It's a good one. We've started on kind of building those buckets. Those are essentially API calls. They are the kind of the most obvious thing to do in terms of representing that. We're looking right now, in fact, we have a project underway to do a similar thing with things like Benchling and other LIMS, SRA as well. So that's kind of already underway. We're just basically picking off the pieces that we see the most users. And so if you do have particular databases or things that you want to access, let us know, and that's kind of something that we can kind of prioritize as we build it out. There's a slight, I guess, a concept of like the kind of data source, and that we have files, which are typically used in bioinformatics, as well as kind of more structured data, which you would think of in databases. And we're still kind of working out exactly how those things live side by side and how you can kind of manage those.
I've got a question from Stav again. Does Data Explorer work for on-prem file systems?
Currently not, but if familiar with how the execution works of pipelines, we have an agent, and that agent essentially is able to retrieve the information and execute the pipeline via a connection with the application. So the idea is to do something very similar with this. So it's kind of early days in that, but it's definitely our intention to build that out.
I've got a question here from Adam. He's asking about the Data Explorer. He says, can I use platform without allowing Data Explorer access to my buckets?
You can, so I guess there's two parts to that. You can hide buckets. That means that they're basically not accessible to people. But then you can also control the access of the buckets given the credentials. So this is something we'd recommend, is that you look at the credentials which you are giving to the application, and then really determine what buckets are available and kind of go from there. Very much kind of standard cloud security policies, but obviously kind of making them accessible.
Personal follow-on from that. I mean, is it possible for someone to set up access for other users within Tower to certain buckets without them knowing the credentials?
Yeah, absolutely. So it's all driven from the security boundary of the workspace. It sounds very technical, but it basically means whoever's in your workspace, you define what they can do. You can define what they see.
We'll try and skip a couple, sorry, but a couple of questions about Data Studio before we wrap up. Abhinav asks, which languages would be available within the Data Studio? Can we build the custom environment container as a basis?
That's a great question, yeah. So we've designed it, obviously, to begin with the most ones which we see the most usage of, but in essence, it is general containerized compute. Nextflow is a single container. It's created and it launches multiple containers into the environment. This is really even simpler in some sense. It's a single container execution into that environment, very much like the head job that you run. And the way that we're thinking of building this is that you can run any container compute. So in the case that I'm showing, it's the kind of the obvious ones, but there's no reason that if you have your own customized environment that you want to spin up, if you want to have, for example, an R-Shiny application, or you want to have some kind of custom compute that you want to run as a service connected to a database, it's all very, very much part of the plan.
One last one. Can you write an ad hoc Nextflow pipeline in the VS Code instance and then run the pipeline directly without a GitHub repo?
Yes and no, you can do it, but it's not very easy, I would say. So this is one, going back to my diagram before, I'm a little bit cheating here, to be honest. Like, this is the kind of goal where I want to be developing, like, wherever I am and be able to do that really quickly. At the moment, it still goes through a Git repository. The way that we're thinking of fixing that is thinking of things like VS Code extensions, which can connect with API directly. So you can imagine pushing to a Git repo, and then basically using the Seqera platform API to have an extension which will kind of launch that and remove a lot of the faff around that.
Okay, thanks very much, Simon.
Awesome, thanks so much.
Okay, thank you, everyone. Very, very pleased to invite onto stage today Eric Garrison, who's our keynote speaker for the event. It's an extremely difficult job to pick a keynote speaker for a conference like this, and lots of names were proposed, but I'm really delighted that, Eric, you're able to join us today. It's a real privilege to have you here, and I'm really excited to hear what you have to tell us. So I'm going to let you do your own intro pretty much.
Okay.
and thanks very much.
All right, thanks, Phil. And thank you all for having me here. It is a pleasure to get to give a keynote at to a conference that's representing such a diverse combination of people working in biology and biotech. And I guess the purpose of this talk is to look a little bit into the future about what happens with the kind of tooling and methods we're working with. And by tooling, I mean everything from the sequencing to the bioinformatic analyses. I guess I'll give a little introduction of myself. I'm an assistant professor at the University of Tennessee, Health Science Center, which is in Memphis. And I've been working in biology for about 10 to 15 years. It's hard to say exactly how much was biology and how much was software tooling. But I basically came into it from following this belief that you should pursue the thing that's most weird and open in front of you. And that effectively led to this place. So first off, I want to give a little bit of technical background, because a lot of the things in the talk won't make sense if I don't provide a background to what pangenomes are and how we work with them. And then I'll talk about human pangenomics. So that's where we have developed a human pangenome that's designed to be an extension and growth and evolution of the reference system that many of you are probably familiar with. And then finally, there's a bit of biology that comes into this, because that system lets us see new things, and they're really interesting. So I'm really happy to get the opportunity to share them with you today. So the first, well, I guess I can give you a little more motivation to this. So when I first started working on actually looking at small variation against reference genomes, so I worked on a variant caller called FreeBayes, I always thought that the most kind of fascinating future direction would be this problem of how do you understand variation between complete genomes? Like, what happens when assembly is solved? Your default is you just like know the genome of an organism, of an individual, of a cell, perhaps. How do you think about the variation between them? Because it's not just all point mutations, right? There's other stuff going on. And so the concept of a pangenome gets at that. I guess to the base level, a pangenome is just a collection of assemblies. So here, this model here, we have 4 genomes, and there's a maternal and a paternal haplotype for each. So they're diploids. And there's this kind of schematic representation saying, if you kind of align them against each other, you'd see that there's a deletion here, there's an insertion there, there's a point mutation somewhere else. And the idea is that those independent assemblies on their own, they aren't really enough to understand what's going on. So we have to put them into a model where that explains both their similarity and their differences. And a standard approach to do that now is this thing called a genome graph, variation graph, pangenome graph. I developed this concept of a variation graph. It actually combines the sequence graph with the genomes that walk through it. And so you can look at the graph and you see the annotations of the genomes through it and understand the variation between them. And so this is a cool thing because it's a compression of those other genomes. So maybe you could use it as a reference system on itself. You could align reads to it. You could think about variation in the population with it. And you can also visualize it. It's a human readable interface into variation between complete genomes. And so, yeah, this squiggle diagram at the bottom is kind of important because there's other ways of looking at this. This is one of them. So here you've got this deletion. So you could skip over a bit of sequence, the CAG, and we could represent that as well as a sort of string diagram where you've got a loop that you could go through, a bubble, or you could skip it. And that's exactly symmetric with an insertion. Indels they can go either way. They both look the same way, basically. But a point mutation, you would have an alternative. So you can go through a G, or you can go through A. And so you get a little bubble like you see on the right-hand side. Then this is a variation graph. This really represents the data model that we're working with because you've got these paths that walk through those sequences. And if you follow the path and you read off the sequences on the nodes of this graph, then you get the genome that is being represented. You get the haplotype that's represented by it. So the idea with this model, the variation graph model, is that you've got alleles or sequences, and you have genomes going through it. And this puts all the genomes and all the sequences in them in relation to each other. So it's basically a solution to this fundamental problem in bioinformatics, which is how do you represent everything and understand how everything relates to everything else? And it's this sort of ultimate bioinformatic object. It's very cool. And I spent about the last 10 years pursuing this topic, trying to first developing the model and then now applying it to data in the context of humans in particular. So the backstory here as well is that it's no longer a dream that you can have a complete genome assembly. Now it's just a matter of a bit of funds because we have there's been this revolution in nanotechnology. We can see single DNA molecules and we can see them with very high precision. And in fact, that's represented by this PacBio system, right? So in Pacific Biosciences, you can observe a single molecule and you can observe it many times through a rolling circle amplification of the molecule. And those many observations let you get to very high accuracy. So you can have reads that are incredibly accurate. And that accuracy and the length of the reads is enough to sort of span most of the repeats in the genome, start to resolve everything, including very complicated regions. But not quite everything, because you also have stuff that's so long and so repetitive that it's very hard to disentangle. And so the ability to see extremely long molecules through Oxford nanopore sequencing, that also, the combination of these two allows us to make complete assemblies, basically without trouble. And so it's becoming reality. Now we have this problem. We get lots of assemblies. How do we understand them? And this is also leading to the development of new resources. Because we can make these assemblies, now there's obvious push to extend the human reference so that it's not just one genome, but represents the diversity of the human population. And so I'll go over some results from this project. The Human Pangenome Reference Consortium, or HPRC, has been funded by the NIH in the United States to develop a reference system that is designed to replace the human reference and extend it. And we get a lot of authors, a lot of different interests, people working in production, people working in ethics, people working in sample collection, and people working downstream of assembling the genomes and people working downstream of them. And we're mostly represented here. So what is, we made a draft, right? So it's our first iteration. There'll be several iterations. The ultimate goal is to have something like 300 to 500 complete genome assemblies. This first one is just 47. And it has a very strange composition because we needed to use something called trio-based assembly to make the assemblies. We wanted to be able to resolve every haplotype in every individual. So every maternal chromosome, every paternal chromosome, we wanted to separate them. And to do this, we needed trios. So we had to take cell lines, and we had to have cell lines, by the way, because sequencing technology works best on cell lines. And so we took 1,000 genome cell lines that had trios and had low cell line passage count and high diversity and maybe had some unique alleles because they were more drifted from the center of human diversity. So that we ended up with this distribution that's mostly genomes from Africa and the Americas with some from East Asia. You know, it's kind of a strange mix, but I think in the future it'll be more balanced because of some of the requirements. We realize we can relax. So we get these 47 assemblies, and they approach the quality of the human reference. The Genome Reference Consortium's build, or H38, or I think it's build 19. It is like pretty remarkable what the sequencing technology allows us to do. So I'm skipping over the whole assembly part just to show that these are really good, and you can see that from this plot because it's showing you a bar that goes from 3 gigabase down to 0, and it's mostly green. And the green means that when we map reads from the assembly, when we map reads from the individual back to the assembly, it looks normal. It looks like it's properly haploid. You know, we get both chromosomes. It's not collapsed. The coverage isn't weird. Only a small fraction, less than 1% of most assemblies, has any obvious issues based on this metric. And so they're really fantastic quality. And the contigs from these, they span whole chromosome arms, typically. So we're able to see really, really large portions of the genome that were invisible pretty much before this project. We'd seen, in some cases, one version in the T to T, that this is a complete human genome reference that was established recently. I'll mention that later. It's described over here on the right, but now we're seeing variation in those regions, right? So given these assemblies, we then wanted to make graphs to describe the variation in all the genomes. And there's 3 methods we used. The first is called minigraph. Minigraph is a progressive approach. It starts with one reference, and it layers things on top of that. And every layering adds a bit more sequence, makes a new graph, and then a new sequence can be aligned against that. So you basically take a series of assemblies, you order them in some way, and you do this sort of progressive alignment. And the result of it is actually that you get a model that's quite simple, which is very nice to work with, because it only encodes structural variation. So any new large sequences or large variation gets encoded in it without the difficulty of thinking about all the small variants, all those SNPs and indels that might each introduce new bubbles, new nodes, make your graph bigger and harder to work with. And so it has this very nice hierarchical kind of model as well that links everything back to the reference. And that can be useful for maintaining compatibility with existing tools. So the minigraph cactus model, this takes the minigraph and it adds the small variants back on top through an alignment. So you build the minigraph and then you align the reads back through that model, or rather, you align the whole genomes back through that model. And then Cactus, which is a kind of multiple whole genome aligner. There's components in that are applied to then rederive the base level alignment. So we can see the SNPs and indels within the model. So this structure matches the minigraph, but then it has the SNPs and indels. It has a small variation inside of it. And so this, it has the same benefits and drawbacks as minigraph. It's progressive, so it's biased relative to the reference, but it does have the same nice hierarchy that links things back to a given reference genome. And then the model that I work on is kind of like the hard mode version. In effect, we align all the genomes to each other directly. And we do this with a technique, the tool is called WF-MASH, but we're linking a very fast homology search algorithm called MASHmap with a very efficient way to do pairwise alignment called the wavefront algorithm. And those two things are brought together in a tool that can scale to really insane problems. Like this matrix that's shown over here, that's actually the alignment of all things homologous to chromosome 6 in primates, in great apes. And it's actually exascale. If you were to count all the bases in that matrix, it's quadrillions. And we can evaluate that very quickly. So we're able to do this comparison that's unbiased, right? So everything against everything else lets us see things without issue. And then this is converted into a graph in an induction step, this algorithm called Seqwish, and then we refine the graph and smooth it. We make it simpler, make it more homogeneous with another tool called SmoothXG. And then there's a lot of downstream things that happen, including visualization, some of which I'll be showing you guys. Oh, I keep doing that. So we did these three methods because we couldn't really agree which one was best. But as a benefit of that was that we had cross-validation. So we had two ways to get to the same result, which was a variation graph that encodes all small variation, all large variation. And so we could extract invariant information. We could do cross-validation with reference-based methods to make sure that this variant calls were correct. And we could then look at the pictures of the pangenome that both of these methods drew. And we basically see that they're quite similar. I won't go too much into the details here, but they're both basically saying that we're adding hundreds of megabase pairs of polymorphic euchromatic sequence. So euchromatic being a kind of active regions of the genome where there's genes that are doing things that we think would be significant, not just repetitive sequences involved in centromeres or other satellite regions. Of those, there's a huge amount. It's almost an endless supply if you were to sequence more and more genomes. But the point is that you're getting 1 to 5 megabase pairs of new sequence per individual. It's a lot, and it's something we've been missing until we embarked on this study. So we use this to then look at interesting loci that are medically relevant. These are two that are quite important. One is the locus that defines the Rh blood group system. This is shown from the mini-graph cactus model, and basically you can see there's these different haplotypes through the locus. There's quite a lot of diversity. That's shown in this figure C, the subpanel C. There's inversions and duplications of some of these genes, and I don't think that before we had these kind of complete assemblies, there hadn't really been this deep survey of this region. And now we're doing it everywhere. Here's another example from the human MHC. This is around HLA-A, and it has a lot of the pseudogenes that are related to it. This is from the PGGB graph, and we showed a bit of both. One interesting thing is that this region around HLA-Y, that's an ancestral sequence. It's quite large. I think it's in the hundreds of kb. And it's not in the reference, actually. It's deleted from the reference. So there are a few sequences like this that were just being left out entirely. They're unique, and we didn't see them before. So this is kind of showing you that you can zoom in from all levels and understand variation at all scales. So at the bottom, you've got one of these kind of string diagrams of the graph. This is the P arm of chromosome 6. And there's a centromere over on the right-hand side, which is this tangled loop, which is quite repetitive. It's kind of sort of a variable number of tandem repeats that makes a loop, and when you build this sort of graph. And then the MHC is this locus. You can actually see it at the scale of the centromere. There's this big bubble. And when you zoom in, that's actually the MHC class 2 gene, HLA-D, and friends, D star, I should say, all the different HLA-D versions and the different haplotypes associated with them, they make this big bubble. And one locus we looked at quite a lot was one called C4A, C4A and B. And that actually forms this kind of loop as well. So there's this particular graph building technique will tend to collapse polymorphic repeats into single loops. So you have one base version of that repetitive sequence, and every genome goes through it multiple times. So this locus is interesting biologically because it's been implicated in schizophrenia risk. There's some contest about that implication, but it's certainly important in the immune system, and it is variable in the human population. People have 1 to maybe 4 or 5 copies typically per haplotype. And one thing that's really nice about this locus is it exposes something that we see because we can make these complete assemblies, which is that there are very unexpected, nonlinear kinds of effects in genome evolution. And what I mean by that, I have to describe a little bit what this figure on the right is to explain. In effect, if you were to take the reference C4A and C4B, and then you take every haplotype through this loop, and you ask, can you deconvolve me relative to the reference? Like, tell me which of those genes I'm more similar to in what order. You get this sort of plot over here on the right. And you can see that some haplotypes have like 1 copy and some have 3, most have 2. But there's something really fascinating that comes out of a few haplotypes, which is that instead of going C4a and C4b, they go C4b and A. And so that means somehow information was getting mixed up in a kind of nonlinear way. And we think, and actually there's a companion paper at the HPRC, that this is being caused by gene conversion. So this is actually a kind of a rampant process in the genome that, homogenization is perhaps the most important kind of recombination going on, but it's been very hard to see if you look at populations with short reads. Okay, so that's a whirlwind tour of a draft human pangenome. So now I want to talk about something new we see, something a part of the genome we couldn't look at before, where we can now understand what's going on. And the subtext is we finished the human genome, right? We actually got a T to T, telomere to telomere assembly of a single human genome. It was made from what's called a hydidiform mole, which is kind of a pseudo-haploid human cell line that had been extracted from this. It's called CHM13. And this actually filled 8% of the human reference that was incomplete. It's mostly centromeric. You can see in this karyotype, the black bars, those are regions we didn't have in the reference. But also the acrocentric short arms, and that's what we'll be focusing on here. Acrocentric chromosome is a chromosome where the centromere is really close to one end. So it has a very short arm and a very long arm. And in humans, all the acrocentrics, they're the locus that codes for ribosomal DNA, which is, in fact, the ribosome biogenesis is the most energetically intensive activity of the cell. So it's a very, very important, and the fact that they all encode for it means that there's this kind of duplication and redundancy, but it also means that these sequences were very hard to resolve because they must be somewhat similar. And so there kind of is a new mystery that comes out of this, and this paper gets into it. I don't have much time to show details of it because I'll have other ways of showing it to you. But when you make the assembly graph out of the hi-fi data for this sample, every single chromosome makes its own component except for the acrocentrics. All 5 of them are involved in one graph component that's isolatable. And that was very suggestive. I think people thought that it was the ribosomal DNA, which is repetitive, that was causing this to happen. But then we also saw it in the HPRC. Our assemblies don't really assemble the ribosomal DNA because it's very, very long repeats and very hard to assemble through. So something else must have been going on. Basically what we're seeing here are, you take the T to C assembly, right, and align each of our HPRC haplotypes against it, and ask when the haplotype switches affinity between reference chromosomes. So switching between reference chromosomes might usually say that seems very unlikely biologically. Like it's weird if you have it, it must be like a cell line artifact, right? You've got two chromosomes fused together or mixed in some way. And we saw maybe 1 or 2 cases like this, but the vast majority of the actual genomes showed this signature of having contigs from the assembly that mixed different acrocentric short arms. And actually contigs, I should note that there's a bit of a jargon from the assembly world. Contig refers to a contiguous set of sequencing reads, like a part of the assembly that you get from overlapping the reads that you're really confident in. And in the HPRC, that's our main currency, because we said, well, we'll take the contigs because we don't really know how they fit together at a higher level. We can see from the perfect overlap of the reads that these are something we can trust. So that's a contig. And we're going to take these contigs. So based on this observation that there's this weird mishmash of things happening, we thought, well, we'll take all the contigs and we'll run the same kind of analysis you see here on the left, where every contig is a node and the edges represent mappings between them. And so here the reference contigs are circled in color, and you can see everything else kind of mixing between them. So we'll do that for all of the contigs and see if, is this happening elsewhere? Like what's basically going on? And so we do this and we get the same kind of figure. This is made by Gephi. It's not like scientifically accurate. It's for human visualization. But the distances are suggestive. And so you see the sex chromosomes form a little cluster, and the acrocentric ones make the same kind of cluster up there that you have the contigs from the Q arm, the long arm of all these chromosomes. They're sort of on the edge. And then in the middle, if you look, it's stuff that maps to the short arm. And it's this big mix of different colors. And the colors we've assigned based on like our best guess of where the content comes from, but they're clearly unreliable, right? Like they're mixed up. And so then we thought to do a quantitative approach to understand what's going on. We applied a community detection algorithm. Actually, we heard a bit about this in the talk about molecular pixelization of cells. So we use this modularity maximizing partition of the same graph I was showing you to break it into parts that are kind of self-contained. And every chromosome or chromosome arm is self-contained except for the sex chromosomes, which you can see kind of at the bottom right. There's the chromosomes at the bottom and the community is up here. So there's multiple things in one community from different chromosomes. And of course, the acrocentrics. And if you look at the actual partitioning, you can see what's going on, that there's this kind of single community representing a lot of the acrocentric cluster. That was very suggestive, but we can make a graph that shows us the exact base pair relationship of these genomes. And so we took the best assemblies from these regions, from the HPRC, and we found ones that found contigs that overlap the centromere. So they go from a part of the chromosome we think is unique, the long arm, into a part that we think is getting mixed up, the short arm. So we take all of these, and of course, what do? We make a graph using PGGB, and that results in this image. But we render it too, right? We make a visualization to get this image. And so the graph has been laid out in such a way that the distances in these 2 dimensions, they match the distances in the contigs that are inside of it, in the haplotypes that are there. And we colored things based on where the T to T chromosomes fit inside of it. And you see that the Q arms are separated. If we were to zoom in on this box, you see in B, there's this very specific relationship between the chromosomes, that 15 is a kind of an outlier, but 21 and 13, they effectively share a centromere, and then everything after it. And the same is true for 14 and 22. And then they come together in this complicated mess. It's actually very hard to disentangle. So we decided to try to disentangle it to understand what's going on, right? Because this is not enough. This is like a kind of population cytogenetic view of what's going on, but it's still just 2 dimensions. It's hard to see. So, is this caused by some kind of homology that's ancestral, or is it something maintained by ongoing recombination? It'd be very exciting. So we untangle the pangenome graph, and that basically involves, for every contig, which are shown here as rows on this plot, at every part of the contig, we're going to ask, what reference chromosome is it most similar to? And so here we've zoomed in, chromosome 13 contigs, they're on the short arm, and you can see the long arm is over here. Everyone matches 13. And then there's the centromere. No one matches anyone, because centromeres are all diversifying, and it's very hard to actually derive this kind of untangling there. And then there's this region, which it's labeled SST1. That's a repetitive sequence in the middle of it. But every single contig is like a patchwork in this region. They're all mixes. And they're specifically mixes of chromosome 13, 14, and 21. And then if you get closer to the ribosomal DNA, there's some other stuff going on with 15. So it starts to play a role. And so to show where this fits on that 2-dimensional plot, that would be this kind of long arm, right? The centromere is this sort of hairball loop where no one is like anyone else. And then this region around SST1 kind of it's really hard to see on the plot, actually, but it's here. And then there's this ribosomal repeat, which we can't assemble through. Only the T to T assembly has any representation of it. And so these regions, actually, I skipped a bit. So we build a kind of entropy metric. We have a quantitative way of doing that that's based on this sort of patchwork pattern you see. Excuse me. And we use that to define what were called pseudo-homologous regions. So these are places where multiple chromosomes are mixing, and they look like they're, hanging out together. And that gives us this sort of intervals across the reference. We get a new annotation type. And we can do this for all the 5 chromosomes. So for 14, you have a similar kind of pattern. Actually, stuff's going on even before the centromere in this case. It likes to match up with 22. 15, like I said, is a bit of an outlier, but close to the ribosomal DNA, it starts to behave the same. 21, it's almost a mirror of what you see in chromosome 13. And 22, again, it's a little bit different than than the three kind of main ones involved in this process, but it also has its pseudo-homologous regions where there's exchanges. So yeah, then we wanted to zoom in on this particular region. It's very interesting, between 13, 14, and 21. The interesting thing about this, the suggestive thing, is that chromosomes 13, 14, and 21 are involved in what are known as Robertsonian translocations. These happen about 1 in every 800 births, and they happen de novo. So there's basically recombination between two chromosomes, and the Robertsonian chromosome is one that mixes two of these acrocentrics. There's two centromeres, and it's lost its ribosomal DNA. And this region looked to be a potential place where that might be happening. One of the suggestive things is that the region is inverted on chromosome 14 in most individuals relative to 21 and 13. So there's a lot going on here. I'm going to just mention that there's a repeat in the middle called SST1, and we looked at the repeat units of it. This repetitive sequence exists on other chromosomes, but if you make a phylogeny of them, every chromosome is like monophyletic, basically. It's its own clade, except for 13, 14, and 21. And so there must be some exchange driving that. And then we dug in with a different way. We looked at older data. There was a fish mapping experiment on Robertsonian chromosomes from 14 and 21. They took probes from the GRC. They took the backs, actually, that were used to make the assembly. And they saw where they fit on the Robertsonian. So if they're present, they're green. If they're not present, they're red. These two plots. And so what we're actually showing is a hypothesis for how the chromosomes match together to make the Robertsonian occur. And given this hypothesis, it's sort of completely consistent with the fish mapping data. So the green bits that you see, and by the way, there's none on 21 because they used the chromosome paint to actually label the Q arm there. It would match recombination in this region. And the fact that you don't find anything on the short arms, this red that you see on the sides, is consistent. And so there's a model of this that's shown below. Basically, if you have this recombination, where 14 is inverted relative to 21, you'll get this microchromosome. It has no centromere, and it'll be lost. And it has the ribosomal DNA on it twice, but it doesn't continue. And then what actually is passed on in the cell line in an individual is this fused chromosome. So this is pretty fun because Robertsonians have been known around for a long time because you can see them using cytogenetic techniques that have been available for 50, 60 years. And so the question about what's going on here was very hard to understand, though, because you can see the result, but you can't see the process, and you can't see the details of ongoing processes you might be maintaining it. So it was brought up in this paper that actually was the first to sequence the centromeric repeats on these chromosomes. They got the sequence of the satellite repeat, and they said, wow, these chromosome 13, 14, 21, they're so similar that probably there's some ongoing recombination exchange between them to maintain that similarity. But that's kind of where the story stops. Everyone says, okay, makes sense. This is another example from the 1970s. Very laborious process. A spermatocyte has been frozen, fixed at what's called pachytene. That's when the maternal and paternal chromosomes have been pulled together and the recombination's occurring. And slices are made through this, and they do an electron micrograph of them, and they're layered them with like sheets of transparency to make a sort of pseudo 3D image, which they then condense into this drawing, in fact. And what's really fun about this is that the nucleoli are these black things. And there's one nucleolus, it has 3 chromosomes on it. And you can guess which they are. They're 13, 14, and 21. So this is an N of 1 thing, but it would look like, this is N of 1, they only did like 2 or 3 of these. So the sample size wasn't very big, but there was a lot of cases where this was occurring in effect. And so they're hanging out together, right? The chromosomes are close physically, which is a precondition for recombination. And this is to show that a direct ascertainment of this is extremely difficult. So if you want to say, find a case where this recombination is occurring, like in the act, what do? You get oocytes where you think that the Robertsonians occur, hundreds of them. They use 300. And then you do FISH to try to color the chromosomes with fluorescent signal. they find one case for 14 and 21 look like they're at pachytene and mixing. So 1 in 300. And that's actually very close to the rate of Robertsonian translocation. So the problem is that to establish, strongly that this is going on, you might have to do 10, 20 times that, and it's just not feasible. So we had the benefit of an amazing resource, human draft pangenome, that lets us look back in time at the human population and do this kind of population genetic analysis of variation of whole chromosomes. And that gives us this insight that there's a region where there must be recombination happening. So there are these pseudo-homologous regions on the short arms of the acrocentrics. And we think that these occur due to physical proximity. Right? The nucleolus is kind of containing the ends of the chromosomes that are associated with the ribosomes. And so they're close together, which gives them the opportunity to recombine. And they have a similar function, too, so they should be similar. So most recombination is actually conversion. About 90% of double-stranded breaks will resolve as a gene conversion event. So this homogenizes the chromosomes relative to each other. We know, based on the fact that Robertsonians exist, that some of these must be crossover, and because how else could you get a Robertsonian chromosome? And so that leads to the synthesis of what's going on and perspective of a region of the genome that was invisible until a few years ago. So we published this alongside the main HPRC paper in Nature in May this year. And it's been an amazing journey to be on, in fact. And I hope it's interesting to you guys to think about what we might see in the future. So I want to thank a lot of the folks who've been involved in this project, in particular, Andrea Guaracino, who did a lot of the hard work required to bring the project to conclusion, and also the whole Human Pangenome Reference Consortium, which made this possible fundamentally. So with that, I'd be happy to answer any questions.
Thank you very much. It was a fascinating story. It was really good. And ties in very neatly. I should little plug for the Boston Summit we have next month where Adam will be talking.
That's
He's
right,
our keynote speaker.
Adam is an important collaborator on this work, and we're actually I didn't mention this, but we're assembling Robertsonian cell lines, and we do have confirmation that that is the region where they recombine. So with Adam,
yeah, we've got lots of questions here. I'm going to pick a couple. Firstly, one from Geraldine. Do you envisage running analyses like variant calling directly on pangenomes as references? And if so, do you think we can still output VCFs, or do we need new formats to represent results?
Yeah, so we can output VCFs. So actually all the work we did in HPRC is sort of ironic. A lot of it feeds back into VCFs against the reference. Even the alignment and variant calling workflows that we presented, they always made a VCF against the reference. So we didn't really explore the full benefit of working on the graph because we lacked the infrastructure, the kind of design, the community infrastructure, of people understanding how to describe variation against the graph. We have models for it, we have data formats, and I think it's completely possible. We have draft versions of ways to do it. But that's interesting. I do want to point out that there also are new opportunities to simplify thinking about variation. So the fact that you have all the alleles that you might be interested in means that you can just jump right into a matrix of allele counts. And that's something very similar to what we work with already in population genetics and GWAS. And I think that could be a way to simplify, kind of cut through a Gordian knot of complex pipelines that we've developed over the past years.
Kind of related to that, maybe unsurprising given the audience, there's an interest in tooling and
Yes.
in building on this.
Yeah.
Abhinav asks, to what extent are current tools compatible for a variation graph-based reference?
Yeah, that's an interesting question. So, in one perspective, nothing works anymore. You say, oh, now I have a graph as my reference, and all is lost. I should rebuild from nothing. And I think that's not accurate because we put all these genomes into that graph. So we actually have many, many frames of reference to work with. And that's part of the basis of our work, was to use those different frames of reference, not just the graph. So any annotations you have against a genome, they can be put into the graph space. They can be injected into that context. And any tools that work on the genomes themselves, they have a natural basis in the graph in that way. So the pangenome is not just graphs, it's really the genomes, and maybe the bigger problem is thinking about just how to work with lots and lots of genomes. Then all the existing tooling we have will continue to work. And we have to do that, and then find ways to harmonize it with the graphs, not necessarily rebuild everything in the graph space.
Last one on tooling. Samuel asks, have you been looking into interoperability between variation graphs and tree sequence data structure used by toolkits like TS-Kit?
Tree sequence data structure, so...
It says it can link different genealogical and phylogenetic trees to different parts of a genome due to the different histories across the sequence.
Right, and this is related to maybe ancestral recombination graphs. I've wanted to find a way to link the two. I guess it's possible in the sense, yeah, based on my previous answer, you know, you've got genomes and you've got ways of working across those coordinate systems. So yeah, in principle, we can do that. We can get variants out of the graph in reference coordinates that can be usable by these existing systems. A deeper integration, though, that would really take someone who loves the two topics. Yeah.
Ricardo asks, how do you represent rearrangements across different chromosomes, and are they independent haplotypes between them? Do you have any ideas on how to do this with polyploids with complex rearrangements?
Okay, well, yeah. So we put a bunch of genomes together, right? So polyploids, no problem, right? You can have one haplotype, you can have 10, it's going to work out. You just have to remember which ones they are. And so remembering which ones they are may be as simple as like naming your sequences appropriately. So there's that part. I think that's okay. What does a rearrangement look like? It's going to be a case, like in the graph, it'll merge different chromosomes together. Because there'll be a sequence that goes from what's typically one chromosome into another. And so you can see that by maybe untangling, or actually by looking at the structure of these two-dimensional layouts or embeddings that can be produced from the graphs. So they sort of naturally work. The only problem is that as you get more and more rearrangements, for example, in a case like in prokaryotes, in bacteria, like E. coli, that have very open pangenomes, then everything is just a hairball. And when you try to look at it in low dimensions, you're completely confused. So yeah, the model represents it, but it may be hard for human eyes to see. Yeah.
Last question from the audience. Hashor says, as a guesstimate, how many human genomes would we need to do to sample to get a good gauge of genome diversity in the human population?
Okay, well, yeah, it's a standard question, right? Like, I guess you can always go down in frequency because we have this rapidly growing population. There's a lot of rare alleles, and so you'll always find something new. And it depends a little bit what part of the genome you want to look at. So what we learned in the HPRC was that the euchromatic sequence is homogenizing, right? And it's under some constraint. So you might have to see fewer genomes to see that. And you would kind of saturate eventually. You'd find most variants, you'd see, maybe not every SNP, but you'd find most large variants or recurrent variants. And this is not true for the satellite sequences and the centromeric sequences. Those are under diversifying selection and kind of, they're like these arms races with each other to be better at being centromeres. And so the more genomes you see, the more centromeres you see. You'll see very big sequences that are, they're not unique in maybe components, like they're made from the same repeat sequences, but the arrangement will be completely unique. And so you could continue doing that forever through the whole human population. But I think that the standard kind of ideas from biomedical informatics apply. So you do 1,000 genomes, you're going to resolve almost anything that you're going to see in anyone. Okay, let's leave the centromeres aside. And you do 100,000, then you really start to cover everything. Maybe almost every base has a mutation, and you learn a lot from that. So I guess this is really an endless process. We've begun. There could be a new pangenome for every population, for the whole human population, for every generation even. Our composition as a species will change over time. So yeah, it's the beginning of something similar to what we're doing, but very expansive in scope.
Right. My final thought is I think it's a really interesting discussion happening on Slack. There's lots of kind of impressions. And my first taste of this was when Simon came and asked about putting a representation of this into multi-QC. And he showed me these beautiful images of string diagrams. And I was like, and I think everyone is kind of sitting there thinking, is this the future of genomics? And you know, is this going to be a watershed moment where we look back on how we used to do bioinformatics and genomics and takes us forward to a more accurate, better representation? I guess, I mean, you're biased, but you guess this is the future.
Okay, it's like, yeah, I mean, it's a certain kind of watershed. I think that confusingly, like, all things will continue. So we'll continue to get short reads because they're super cheap. And we can then use them against this system to do better. But the short reads will exist, right? We still have array data, and it's really cheap because all the things that you got good at doing in the past, they become very low cost, and they have their utility. So I feel like this is just an expansion of the kind of scope that genomics can have, you know, rather than a replacement. And it's not like things that we've been doing in the past become irrelevant.
Thank you very much. Thank you for giving us a glimpse to the future.
Yeah.
Thank you.
Thank you so much. Thank you all.
Okay, so we've reached the end of the session. We're going to finish up with a little announcement that we have relating to the rest of the evening. There'll also be graph genomes if you want as well. It's optional as well there. Okay. So just switch to my slides there. So we're really excited to announce that as part of the America's Cup being a part of Barcelona for the next couple of years, that we're going to be sponsoring the Alinghi Red Bull racing team as official supplier for data analysis, high-performance computing as well. More related to us tonight, though, we will be spending the evening at their base there. I think we can jump to there's a space for a video here where you can watch it, and I will provide the details after that. We can dim the lights and get a 60-second video, I believe.
Get it when I get back. Click, clap, blaster.
So in about 60 minutes, hopefully you'll find it in the event information that you have there. This is the address down there. So this is the Alinghi Red Bull base. One final thing, they have also been really nice and given us, someone will basically win a 9,000 euro bike today. Completely unexpected, to be fair. So when you go in there, you'll get a ticket. You can use that ticket, and we will do a draw that courtesy of the Alinghi Red Bull racing team. I'll put the details here. Thanks so much for a great first session. Really look forward to spending some time and showing you tonight.